studying the very important cell organelle, that is nucleus. So up till now we have discussed about all other organelles. Now we are going to study about the nucleus, which is the controlling center of the cell. So when we are going to stain some cell, that time the spherical bodies which we observe, which are almost at the center, more or less at the center they are present, which are getting stained, and they may vary in their position. This position, which I said central position, it may vary in some cells. Otherwise, the common position is central. That is the controlling center of the cell. So these spherical bodies, we call them as nucleus, which are having the detailed structure like this, where this spherical body is bound by the envelope. We call it as nuclear envelope, which is double membrane structure. So each unit membrane, which is lipoproteinaceous in nature, and that is around about 60 angstrom units in thickness. There is space between these two unit membranes, and this space we call it as perinuclear space, which is about 150 to 300 angstrom units in width, this perinuclear space, and it is filled with fluid. We call it as perinuclear fluid, which is protective in nature. Inside we observe entire volume filled with the fluid. We call it as nucleoplasm. This nucleoplasm, it contains the chromatic network or thread-like structures, which are the DNA, which are going to get packaged with the help of histone and non-histone proteins, forming the chromosomes during cell division. So that is chromatic thread network. And here one more structure we are observing, that is again the darkly stained body, which is present over here. We call it as nucleolus, and this region, which is rich in ribonucleoproteins, it is involved in the formation of the ribosomes. That is nucleolus. At some places we observe pore to the nuclear membrane or envelope to maintain the continuity with the surroundings. Even at some places we observe continuation of the rough endoplasmic reticulum, that is starting from here. While discussing the endoplasmic reticulum, we have discussed about it. That is, it is starting from the nuclear envelope and it goes to the SER, that is smooth ER, and then to the entire cell end, that is the cell membrane. Here at some places you are observing ribosomes on this membrane. Even presence of ribosomes is there on this membrane of the rough endoplasmic reticulum. So it is all about the structural arrangement of the nucleus, which we observe in case of eukaryotic cells. What are the functions which are attributed to this? The functions, that is, first, to control almost all activities of the cells. Entire cell is under the control of nucleus. Second function, it helps in maintenance of the cell, and that is why without nucleus cell will not last longer. Cell will die without nucleus. Third function, again very significant role, that is it carries genetic information in the form of DNA, and that genetic information is responsible for metabolism, development, reproduction, that is all cellular processes or all life processes, they are having the genetic information over here. One more function, that is with the help of ribonucleoproteins. It is involved in the synthesis of ribosomes, which are the very important involvement in protein synthesis. We know ribosomes, they are involved in protein synthesis, and these ribosomes, they are by the nucleus. With the help of ribonucleoproteins, they are synthesized. So it is all about the nucleus, which is the governing center or controlling center of the cell. So it is the eukaryotic feature.